In today's class, we are going to discuss about the methods of artificial vegetative propagation. Previously, we have known about the natural vegetative propagation, which included the reproduction of plants by the stems, roots and leaves. And in nowadays, the farmers and the horticulturalists have developed certain artificial methods of vegetative propagation, and today we are going to know about them. The first method of artificial vegetative propagation is called cutting. This method is applied in propagating plants like sugarcane, rose, China rose, lemon, etc. In this method, what is done is that the stem is cut into small pieces. Okay, just we can see in the picture with the blade, the parent plant is cut into small pieces, and that part should bear an axillary bud, and then after the cut ends are planted in moist soil, and after a few days they will strike roots and that part will grow as an individual plant. This process is known as cutting. The second method of artificial vegetative propagation is called layering, and it is done in mint, rose and jasmine. What is done that a portion of one of the lower branches of the plant with an axillary bud at the node is bent into the ground, and then it is put into the soil, and then it is actually topped with some heavy thing, so that with the weight of that heavy thing, that part of the branch remains inside the ground. And when that part of the soil is watered, after few days, those bent part of the plant will strike root, and then it is cut and it will grow as an individual plant, and it will prosper as an individual plant. The third method is called the grafting, and it is done in rose, mango, guava, etc. The plant receiving the bud or shoot is called the stalk, and the shoot fixed on it is called the scion. And what is done that the plant receiving the bud, which is called or bud or the shoot is called the stalk. For a successful graft, it is important that the cambium layers of the stalk and the scion must come into very close contact, so that the growth may continue. The continuous growth may continue. The grafted points are then bound together with the tape, and then it is topped with some plastic bag, so it does not get attacked with some bacteria or virus or microbes, and to prevent dehydration. In a few days, the new cells develop and a new plant grows, and it will have characters of both the scion and the stalk. The last method of artificial vegetative propagation, and it is the most professional method and is done in the laboratory only. It is called micropropagation. It has a few steps. So we take a plant, and that has a very healthy shoot apex. Now we cut the shoot tip or the meristem. It is the growing part of the plant, so it is actually composed of meristematic tissues. Then we keep the shoot tip or the meristematic tissue into a primary culture. The primary culture is nothing but a solution with the essential nutrients that are required for the growth of the plant. Now, after some days, the shoot development will begin with axillary shoot proliferation. It will grow into size. And after that, the roots will come out. First, the shoot will come out, and then the roots will come out. And ultimately, it will be a small rooted plant. And then we can remove that rooted plant from that primary culture, and then we can pot it into a tub, and it will grow as an independent plant. Now, there are few terms like explant, tissues of the vegetative buds, shoot apex, which is used to start the micropropagation process, that is called the explant. Now, what is callus? The cells grow and divide to form a mass of cell. Like the primary culture, whenever we put the shoot tip into the primary culture, after some time, the shoot development with axillary shoot proliferation, that stage is called the callus. It is just a mass of cell. And plantlet is the small rooted plant that is actually removed from the primary culture and potted into the plant. So explant, callus and plantlet are the three important terms associated with micropropagation.